If we're able to identify a binder for the RBX1 protein, could potentially be a cancer therapeutic.
Hello, my name is Matthias. I'm CTO and co-founder of Liquid, and this is Building at Liquid. Today I'm here with Kaeli.
Hi, I'm Kaeli. I'm one of the machine learning research engineers, and I work on biological applications of LFMs.
Foundation models are all about large language model, text, but there are much more interesting data domains than just text.
Yeah, so I work on a protein language model at Liquid. We're exploiting the long context capabilities of LFMs to train on up to hundreds of protein sequences in a given sample. And then we can hopefully generate proteins that fall along the same evolutionary lineage or have the same function, same structure. And to my knowledge, this is probably the most flexible protein design framework possible. A lot of other protein design approaches are structure-based, so you give the model a geometry that you need to conform to. We're not doing that. It's all sequence-based. So we've had a lot of success with this in silico so far.
So instead of a next token or next text word, we're predicting here the next amino acid, or like?
Yes, exactly. So it is still like autoregression. We're predicting one token at a time, but the tokens aren't natural language characters. They are amino acids.
That's amazing. Like what's the sequence length that you usually pre-train in those models?
Totally depends on the types of proteins that we're trying to design. For now, we've been training at 32K context. And the average protein length in the databases we use is between, it's like a few hundred. So we can pack up to thousands, potentially, hundreds to thousands into our context length.
So during training in the in silico portion, basically, of the protein design, how do you measure like how well the model is fitting or how well the model is doing?
I'm sure you've heard of AlphaFold. So we use that we use a public extrapolation of the AlphaFold algorithm for structure predictions. So we can visualize these structures. We can kind of vibe check. You can also visualize docked structures, so you can see how, if you're designing a binder, how it seems to fit with the substrate you're targeting. So that's all structure-based, but of course we have sequence-based signature scans. It's called interpro scan. That's checking to see if we are aligning with the signatures that we purposefully set in the prefill.
That's great. So with AlphaFold and a lot of the methods that you mentioned to validate in silico, but then the most exciting part is probably like sending it to the lab. So can you also share?
Yes. So we've been working with a biotech company to help us perform assays that directly try to assess all of these different enzymatic functions. So we've sent them some control samples already for something called the PETase enzyme, which is supposed to break down plastic. So once we have good results on the controls and we know that their laboratory environment works for the types of enzymes that we're trying to design, we'll send them our designs, and hopefully we can converge to something extremely successful with that method.
We're not just designing the proteins in theory or on a computer and write a paper about it, but these are actually being produced.
Yeah. Designing PETase enzymes in particular obviously has a lot of real-world value, breaking down plastic. And in a lot of cancers, RBX1 binds with these other proteins to form many copies of this E3 ligase. And that causes the cell cycle to spin out of control because it's destroying other proteins that are very important in the cell cycle. So the cell cycle spins out of control, you get tumor growth. So if we're able to identify a binder for the RBX1 protein that prevents it from forming that other compound that then causes the cell cycle to spin out of control, could potentially be a cancer therapeutic. But that could have definitely real-world impact.